3 years ago, I started noticing something deeply troubling in my emergency practice. I'd see patients come in after their cancer treatments, radiation, chemotherapy, the works. They'd beaten the cancer. Or so we thought. But 6 months later, a year later, they'd be back. The cancer had returned, more aggressive than before. Different patients, different cancer types, but the same devastating pattern. One woman, 73 years old, breast cancer survivor, told me something that stopped me cold. She said, "Dr. Becker, I did everything they told me. I suffered through the chemo, the radiation, lost my hair, couldn't eat for weeks. They said I was cancer-free. So why am I back here 9 months later with tumors in my liver?" That question haunted me. And when I started digging into the research, what I discovered was so fundamentally different from what we've been taught about cancer that it challenged everything I thought I knew about this disease. I'm Dr. Becker, and I've spent 20 years in emergency medicine watching both the acute crisis and the slow decline that leads to them. What I'm about to share comes from reviewing hundreds of studies following the work of researchers who've been investigating cancer metabolism for decades and watching how this plays out in real patients over 65. Because here's what nobody's telling you. The food you're eating right now, the meals you think are healthy, could be directly feeding cancer cells in your body. And the treatments being used at every major cancer center in the country might be making the problem worse, not better. The scale of this is staggering. Nearly 1,700 people die from cancer every single day in the United States alone. We're spending billions on treatments that haven't moved the survival needle in any meaningful way for the majority of cancers. But there's a completely different approach, one based on how cancer cells actually get their energy that's showing remarkable results in the patients who've tried it. Today, I'm walking you through the complete investigation. You'll see the research that's been ignored for nearly a century. Understand the biological mechanism that explains why cancer grows out of control. And by the end, you'll know exactly which foods are feeding cancer cells and which approach could starve them without the devastating toxicity of conventional treatments. Think of cancer not as a genetic curse, but as an energy problem. Your cells have switched from one fuel source to another. And that switch is what's driving the disease. Once you understand that, everything changes. Before we go deeper, if you're finding this valuable, a subscription helps me continue investigating topics like this that could genuinely save lives. No pressure, just appreciate the support. The research trail that changed my perspective started with work done nearly a century ago by a German scientist named Otto Warburg. In the 1920s and 30s, Warburg made a discovery that should have revolutionized cancer treatment. He found that cancer cells have a fundamentally different metabolism than normal cells. Here's what stopped me when I read his work. Warburg discovered that if you take a rat with a large tumor and give that rat cyanide, the rat dies instantly, but the tumor keeps growing. Think about that for a moment. Cyanide kills by blocking cells from using oxygen to make energy. It shuts down what's called oxidative phosphorylation, the normal way your cells generate power. But the tumor doesn't die. Why? Because cancer cells aren't using oxygen for energy. They're using an ancient fermentation process that doesn't require oxygen at all. Warburg's hypothesis was this. Cancer begins when the mitochondria, those tiny power plants inside your cells that use oxygen to make energy, become damaged. When that damage happens chronically, not all at once but gradually over time, cells are forced to switch to a primitive backup energy system, fermentation, the same process that bacteria used billions of years ago before oxygen existed in Earth's atmosphere. And here's the critical part. The mitochondria don't just make energy. They also regulate whether a cell stays differentiated doing its specialized job or starts dividing uncontrollably. When mitochondria fail, cells lose that regulation and begin proliferating without restraint. That's cancer. For decades, this work was largely ignored. The field of oncology went all in on the theory that cancer is primarily a genetic disease caused by mutations in DNA. But a 2024 meta-analysis published in the International Journal of Molecular Sciences analyzed data from over 12,000 individuals across 32 separate studies. What they found validates everything Warburg said. People with chronic fatigue syndrome had 30 to 50% fewer mitochondria in their cells compared to healthy controls. Type 2 diabetics had 30% fewer mitochondria in muscle and pancreas cells. Heart disease patients showed 40% fewer mitochondria in heart muscle. Alzheimer's patients had 30 to 50% fewer mitochondria in brain neurons. And cancer patients, same pattern, massive mitochondrial dysfunction across every cancer type studied. Do you see it? Every major age-related disease has the same underlying problem. Your cellular power plants are dying off or shutting down. When mitochondria fail, organs can't function properly. Your brain can't think, your heart can't pump efficiently, your metabolism can't burn fat, and cells start growing out of control because the very organelle that regulates growth has become defective. But here's where it gets even more interesting. A study from the Buck Institute for Research on Aging followed 847 adults over 65 for 8 full years, measuring something specific called mitochondrial density using advanced cellular imaging. The participants who maintained or increased their mitochondrial count showed 67% slower cognitive decline, 42% better physical endurance, and 53% lower rates of age-related disease overall. They weren't just measuring how people felt. They used standardized neuropsychological testing and objective physical performance measures. This was hard science, proving that cellular energy capacity directly determines how fast you age and whether you develop cancer. Now, here's what really matters for understanding how to fight cancer. Warburg clearly stated that if oxidative phosphorylation, energy production using oxygen, is damaged too quickly, the cell just dies. A dead cell can never become a cancer cell. So, the origin of cancer requires chronic gradual damage to mitochondrial function. The damage has to happen slowly enough that the cell has time to upregulate an alternative energy source to replace the lost energy from oxidative phosphorylation. That alternative source is fermentation, and fermentation requires specific fuels. This is where everything clicks into place. Once I understood that mitochondrial failure was driving cancer, I had to know what happens at the biological level. Why do mitochondria fail? The answer involves 5 interconnected systems that explain not just cancer, but why it's become epidemic in modern society. System 1 is oxidative stress damage. Your mitochondria are like tiny furnaces burning fuel to create energy. Every time they burn fuel, they create exhaust in the form of free radicals that damage the mitochondrial structure itself. In your 20s and 30s, your body produces enough antioxidants to neutralize most of this damage. But after 60, antioxidant production drops by 30 to 40%, while free radical production actually increases. A 2023 study in the journal Aging Cell measured oxidative damage markers in mitochondria across different age groups. By age 70, mitochondrial DNA showed 3 times more oxidative damage than at age 30. That cumulative damage is why energy crashes as we age. System 2 is chronic inflammation, what researchers call inflammaging. When your immune system is constantly activated by poor gut health, processed foods, environmental toxins, or chronic stress, it releases inflammatory signaling molecules called cytokines. These cytokines trigger a process where damaged mitochondria are cleared out and recycled. That's normally healthy. But when inflammation becomes chronic, this process goes into overdrive and starts destroying healthy mitochondria too. A study in the journal Immunity tracked inflammatory markers and mitochondrial counts in aging populations. Every 10-point increase in inflammatory cytokine levels correlated with an 8% reduction in mitochondrial density. Your immune system trying to protect you inadvertently destroys your energy factories. System 3 is sleep deprivation. During deep sleep, your body activates mitochondrial biogenesis, the actual creation of new mitochondria to replace those lost during the day. But this only happens during deep slow-wave sleep. A study published in Science Advances used specialized imaging to watch this process in real time. Mitochondrial repair and replication peaks between 1 and 3 a.m. during deep sleep phases. When participants had their deep sleep disrupted, even if total sleep time stayed the same, mitochondrial biogenesis dropped by 47%. If you're not getting quality deep sleep, you're not replacing the mitochondria you're losing. You operate at a deficit that compounds every day. System 4 involves something called NAD+, nicotinamide adenine nucleotide. This molecule is absolutely essential for mitochondria to produce energy. Think of it as the ignition key. Without enough NAD+, mitochondria can't convert food into usable energy, no matter how much you eat. Here's the devastating part. NAD+ levels drop by 50% between age 40 and age 60. A 2022 clinical trial published in Nature Metabolism measured this decline and tested whether supplementing NAD+ could reverse mitochondrial dysfunction. Participants who increased their NAD+ levels showed measurable improvements in energy production within 6 weeks. Mitochondrial efficiency increased by 31%. This wasn't subjective feeling better. They measured actual ATP output, the energy currency your cells use. System 5 is nutrient deficiencies in the specific coactors mitochondria need to function. B vitamins, coenzyme Q10, magnesium, iron, and several others act like the oil and spark plugs in your cellular engine. A comprehensive study in the American Journal of Clinical Nutrition analyzed micronutrient status in adults over 65. 74% were deficient in at least 2 mitochondrial coactors. 41% were deficient in 4 or more. Even if standard blood work looks normal, if you're missing these specific nutrients, mitochondria cannot produce optimal energy. Now, here's what nobody talks about. These 5 systems don't just add up, they multiply each other's effects. If you have oxidative stress plus inflammation plus poor sleep, the damage isn't 3 times worse. It's exponentially worse because each system amplifies the others. That's why some people decline rapidly while others maintain energy. It's not genetics. It's the cascade effect of multiple systems failing simultaneously. For decades, we've been told that cancer is fundamentally a genetic disease. Mutations in DNA cause cells to grow out of control, and we need to target those mutations with drugs. I believed this myself for years. Then I read a meta-analysis that challenged everything. The researchers separated people who aged chronologically, just getting older in years, from people who aged biologically, showing actual cellular decline. Chronological age had almost no correlation with mitochondrial health or cancer risk, but biological age, measured by inflammation markers, oxidative stress, and metabolic health, predicted mitochondrial density with 92% accuracy. In plain English, the number of years you've lived doesn't determine whether you get cancer. How well you've maintained your cellular health determines that. Here's the paradigm shift. The mutations we see in cancer cells, the ones the entire field focuses on, they're not the cause, they're the effect. When mitochondria become dysfunctional, they throw out reactive oxygen species, free radicals that are carcinogenic and mutagenic. These radicals damage proteins, lipids, and DNA. The mutations that oncologists obsess over, the p53 mutations, the EGFR mutations, all of them are downstream effects of mitochondrial damage. The entire cancer field is focusing their energy on downstream stuff that's largely irrelevant to the majority of cancers. Here's the critical question nobody's asking. If cancer cells have damaged mitochondria and can't use oxygen for energy, what are they using? The answer is fermentation. And fermentation requires specific fuels. Glucose, that's sugar, is the primary fermentable fuel. But recent research has identified a second critical fuel, the amino acid glutamine. Cancer cells ferment both glucose and glutamine to generate the energy they need to grow and divide. This is exactly what Warburg predicted, though he didn't know about glutamine fermentation at the time. Think about what this means. Normal cells in your body can use oxygen to burn fatty acids and ketone bodies for energy. They're metabolically flexible. Cancer cells have lost that flexibility. They're locked into fermentation, which means they're absolutely dependent on glucose and glutamine. If you remove those two fuels from the microenvironment around a tumor, you create a situation where normal cells thrive while cancer cells are placed under enormous metabolic stress. This is where we get to the foods that feed cancer and the approach that could starve it. So, I've shown you the research explaining that cancer is fundamentally an energy problem, not primarily a genetic one. I've explained the mechanism. Damaged mitochondria force cells to rely on fermentation, which requires glucose and glutamine. The question everyone asks is, Doctor, what do I actually do with this information? Because understanding the problem only matters if there's a solution you can implement. After reviewing hundreds of studies and following the clinical work of researchers using metabolic approaches, I've identified the specific dietary strategy that targets cancer's energy supply while protecting and even enhancing the health of your normal cells. This isn't about deprivation or suffering. It's about precision, giving your body the fuels that normal cells can use while starving cancer cells of the fuels they require. Priority 1, and this is the foundation everything else builds on, you must dramatically reduce glucose availability to cancer cells. Glucose is the primary fermentable fuel driving tumor growth. Every time you consume foods that spike blood sugar, you're directly feeding cancer cells. Here's what the research shows matters most. Eliminate or drastically minimize refined carbohydrates, sugars, and high glycemic foods. That means no table sugar, no high fructose corn syrup, no honey, no agave, no artificial sweeteners that spike insulin, no fruit juices, even natural ones, no sodas, not even diet sodas, no white bread, white rice, white pasta, no crackers, cookies, cakes, pastries, muffins, donuts, no breakfast cereals, even the ones marketed as healthy, no granola bars, energy bars, protein bars with added sugars. But it's not just obvious sweets. Many foods you think are healthy spike blood sugar dangerously high. Whole wheat bread raises blood glucose almost as much as white bread. Most commercial yogurts have more sugar than ice cream. Dried fruits are concentrated sugar bombs. Even certain vegetables like potatoes, especially in processed forms, create massive glucose spikes. A baked potato has a glycemic index of 95, nearly identical to pure glucose. Here's what you should be eating instead. Non-starchy vegetables should be the foundation. Leafy greens like spinach, kale, arugula, chard. Cruciferous vegetables like broccoli, cauliflower, Brussels sprouts, cabbage. These provide fiber, micronutrients, and compounds that support mitochondrial function while having minimal impact on blood sugar. Healthy fats become your primary energy source. Extra virgin olive oil, avocados, nuts like walnuts and pecans, seeds like chia and flax, wild-caught fatty fish like salmon, sardines, mackerel loaded with omega-3 fatty acids that reduce inflammation and support mitochondrial membranes. Moderate amounts of high-quality protein, grass-fed beef, pasture-raised poultry, wild-caught fish, eggs from pastured hens, and here's one most people miss, but is incredibly powerful. Organ meats, liver, heart, kidney. These are the most nutrient-dense foods on Earth for mitochondrial coactors. If you can't stomach eating them, desiccated organ supplements from grass-fed sources work. 3 to 6 capsules daily provides the B vitamins, coenzyme Q10, iron, and other nutrients mitochondria need. The goal is keeping your blood glucose stable and low, ideally between 70 and 90 milligrams per deciliter. When you achieve this, your body shifts into producing ketone bodies, water-soluble breakdown products of fat that your liver makes when carbohydrate intake is very low. Ketone bodies are an alternative fuel that normal cells with healthy mitochondria can use efficiently. Cancer cells cannot use ketone bodies because they require functional mitochondria to burn them. This is the metabolic switch that makes everything work. A study published in Cell Metabolism tested this approach in adults aged 65 to 80 who were sedentary, not athletes. They followed a protocol involving calorie-restricted ketogenic eating for 12 weeks. Mitochondrial content increased by 69%. Their cells literally created more power plants. Energy improved dramatically. Cognitive function sharpened, and markers of metabolic health transformed. But here's the critical nuance for cancer specifically. Ketogenic diets alone do not kill cancer cells. Ketones are for the benefit of your normal healthy cells. They make those cells more resilient, more energized, better able to withstand stress. Cancer cells don't benefit from ketones because they can't metabolize them, but you still need to actively restrict the fuels cancer cells do use, glucose and glutamine. Priority 2 is targeting glutamine. This is where it gets more complex because you cannot eliminate glutamine through diet alone. Glutamine is the most abundant amino acid in your bloodstream. Your body makes it, and it's in virtually every protein-containing food. For cancer patients, specific pharmaceutical interventions are required to restrict glutamine availability to tumors. The drug DAWN 6-DAZO5-OXO-LN-Norlucine blocks glutamine utilization pathways. In preclinical studies, combining glucose restriction through diet with glutamine restriction through targeted drugs created a synergistic effect that placed tumors under severe metabolic stress while normal cells remained healthy. This is what researchers call the press-pulse strategy. The press is the constant metabolic pressure from the ketogenic diet, keeping glucose low. The pulse is the intermittent use of specific drugs in low doses that target the glutamine pathway and other cancer vulnerabilities because the tumor is already under stress from glucose restriction. Much lower doses of drugs are needed. This reduces toxicity dramatically while increasing effectiveness. Priority 3 is enhancing mitochondrial function in your healthy cells. Remember, cancer is fundamentally about damaged mitochondria. So while you're starving cancer cells, you simultaneously want to support and regenerate healthy mitochondria in all your normal cells. Several nutrients have strong research backing for direct mitochondrial support. Coenzyme Q10 in the ubiquinol form, the active form your body uses, 200 mg daily with a fat-containing meal for absorption. A meta-analysis of 17 studies showed CoQ10 supplementation reduced fatigue scores by 32% in adults over 60 with mitochondrial dysfunction. It's essential for ATP production and acts as a powerful antioxidant protecting mitochondria. NADI plus precursors, specifically NMN or nicotinamide mononucleotide. Doing is 250 to 500 milligrams taken in the morning on an empty stomach. Studies show blood NAD+ levels increase by 38% within 6 weeks at this dose. Some people combine NMN with resveratrol 100 mg of transresveratrol. Resveratrol activates sirtuins enzymes that regulate mitochondrial function, and sirtuins require NAD+ to work. You're hitting the pathway from both directions. A clinical trial showed this combination increased mitochondrial efficiency by 41%. Alpha-lipoic acid at 600 mg daily split into 2 doses. This is one of the few antioxidants that's both water and fat soluble, meaning it reaches every part of the cell, including mitochondria. Studies demonstrate it reduces mitochondrial oxidative stress by 47%. PQQ pyrroliquinoline quinone at 20 mg daily. This compound stimulates mitochondrial biogenesis, the creation of new mitochondria. Studies show PQQ supplementation can increase mitochondrial number by 20 to 30%. The richest food source is natto, fermented soybeans consumed in Japanese blue zones, where people routinely live past 100 with remarkable energy. One serving provides more PQQ than any supplement. Priority 4 involves specific foods that contain unique molecules directly enhancing mitochondrial function. At the top of that list are pomegranates. They contain compounds called elagitanins that gut bacteria convert into urolithin A. This molecule stimulates both the removal of damaged mitochondria and the creation of new ones simultaneously. A randomized control trial gave older adults either pomegranate extract or placebo. After 4 months, the pomegranate group showed 22% improvement in muscle endurance and 17% better mitochondrial efficiency. 1 cup of pomegranate arils daily, or 8 ounces of pure pomegranate juice, provides therapeutic amounts. Berries, especially blueberries and blackberries, contain compounds that reduce mitochondrial oxidative stress. One study showed 1 cup of blueberries daily increased mitochondrial density by 12% over 12 weeks. Dark leafy greens provide magnesium, absolutely critical for ATP production. 3 cups raw or 1 cup cooked daily. Extra virgin olive oil contains oleoenthal, which reduces mitochondrial inflammation. 2 tablespoons daily used raw on salads or vegetables. Priority 5 is lifestyle interventions that trigger mitochondrial biogenesis. The single most powerful non-dietary intervention for creating new mitochondria is high-intensity interval training. A landmark study in Cell Metabolism tested this in adults aged 65 to 80, sedentary individuals who weren't exercising. They followed a specific protocol: 30 seconds of elevated effort followed by 3 minutes of recovery, repeated 6 times, done 3 times per week. After 12 weeks, mitochondrial content increased by 69% in older adults compared to 49% in younger adults. The mechanism is this: when you push your cardiovascular system hard enough to slightly exceed comfortable capacity, you trigger AMK activation. EMPK is the master switch for mitochondrial biogenesis. It signals cells that energy demand is high and new mitochondria need to be built. The sweet spot for most seniors is perceived exertion of 7 out of 10 during the intense intervals. You should be breathing hard enough that conversation is difficult but not impossible. This works on a recumbent bike, elliptical, or even brisk walking intervals. If you're building up, the key is consistency. 3 times weekly, non-negotiable. Priority 6 is optimizing sleep for mitochondrial repair. Your mitochondria repair themselves during sleep, specifically deep sleep. Chronic sleep deprivation leads to oxidative stress and damages mitochondrial DNA. Studies show sleep deprivation triggers brain inflammation, and mitochondria in brain cells are especially vulnerable for cancer patients or anyone trying to prevent cancer. Sleep optimization is not optional. Bedroom temperature between 65 and 68 degree F. Blackout conditions genuinely dark, where you cannot see your hand. Asleep, not just in bed by 10:30 p.m. to catch the first deep sleep cycle. Stop all fluid intake 3 hours before bed to avoid waking to urinate, which destroys deep sleep cycles. Priority 7 is managing chronic inflammation to stop mitochondrial destruction. Remember, chronic inflammation triggers excessive destruction of healthy mitochondria. The foundation is eliminating inflammatory foods. That means drastically reducing or eliminating seed oils like soybean, corn, canola, and cottonseed oil. These are in virtually every processed food and restaurant meal. They're loaded with omega-6 fatty acids that drive inflammation when consumed in excess. No fried foods, minimal processed foods with chemical additives. What to emphasize instead are omega-3 rich foods, wild-caught fatty fish 3 times weekly, probiotic foods like unsweetened yogurt, kefir, sauerkraut to support gut health. Since 70% of your immune system lives in your gut, prebiotic fibers from vegetables like asparagus, garlic, onions, artichokes, specific anti-inflammatory compounds like curcumin from turmeric, but you need black pepper to absorb it. $1,000 mirrors curcumin with 5 milligrams piperine daily ginger fresh, or supplemented at 1 gram daily. Now, here's what's critical to understand. This isn't just about individual interventions. It's about creating a complete metabolic environment that places cancer cells under stress while making your healthy cells more resilient. When you combine glucose restriction through diet, enhanced mitochondrial support through targeted nutrients, inflammation reduction, optimized sleep, and strategic exercise, you create a situation where cancer cells struggle to survive while your normal cells thrive. But there's something else we need to address. What about conventional cancer treatments? This is where things get controversial, but the science is clear. A study analyzing outcomes across multiple cancer centers found something disturbing. When glioblastoma patients, that's an aggressive brain cancer, receive standard radiation therapy, the treatment dramatically increased glutamine levels in the tumor microenvironment. Radiation was giving cancer cells a massive influx of one of their 2 critical fuels, High-dose steroids given to control brain swelling from radiation spike blood glucose to very high levels. So the very treatments meant to kill the cancer were simultaneously feeding it with both glucose and glutamine. This helps explain why glioblastoma has such dismal survival rates despite aggressive treatment. Standard of care for GBM kills more than 90% of patients before 5 years. I'll see you in the next